let's just talk briefly about the eye. Of course, everyone is used to the view on the left side of the eye. That's the view you see. If you were to take a cross-section, this is what it looks like. The light comes in through the cornea, which is the clear window on the front. It gets additional focusing by the lens inside your eye and ends up hitting the retina. And the retina, if we want to sort of zoom in there, that's actually where a lot of the action is for a lot of the eye diseases that we have. Back in that retina, there are a variety of cells. There are rods and cones. Those are the photoreceptors that let you see in the dark and in the light with color, respectively. There's a lot of processing that happens in the retina before those visual signals get sent to retinal ganglion cells, and they carry all that vision from your eye back to your brain down the optic nerve. And that's another source of degenerative disease. So what goes wrong sometimes? Well, the photoreceptors, the rods and cones, and the support cells, the retinal pigment epithelium, they degenerate in diseases like age-related macular degeneration, or AMD, very common in the aging population. And when we look into the retina, we see the damage happening right in the center of where your vision is supposed to be at its best. On the other end, the retinal ganglion cells and their fibers carrying information down the optic nerve, they degenerate in glaucoma, another exceedingly common cause of vision loss. And when we look into your eye, and we can actually see the optic nerve when we're screening inside the eye and see whether it looks like glaucoma and pick that up. Another really, unfortunately, increasing cause of vision loss in our country and other developed countries is diabetic retinopathy. And diabetes actually greatly damages all of the cells in the retina. And again, when we look into the eye, we can tell whether a diabetic is having good blood sugar control and looking pretty healthy, or having trouble with their blood sugar control and accumulating damage. So the fundamental problem that we have in these diseases, once we get outside of trying to prevent them, is that there's no regeneration or repair or replacement in the retina in injury or disease. And that's our fundamental issue of why so many of these degenerative eye diseases lead to permanent vision loss. And these are significant. You know, if this might be your normal view of a couple of children in the backyard, with macular degeneration, it really affects the center of your vision early. With glaucoma, it affects the edges of your vision early and then narrows into the center late in the disease. And these create a huge societal burden. In the developed world, age-related macular degeneration is the #1 cause of irreversible blindness in the developed world. Diabetic retinopathy, because diabetes is increasing in the developed world, that's really catching up to macular degeneration. If you look worldwide, glaucoma is actually the #1 cause of irreversible blindness in the world. And the cost to our state, to our society, to the world is enormous. In the United States, they've added up the cost in billions of eye disease. And when you include the direct costs, what does it take to treat patients, as well as the indirect costs, people who, because of their poor vision, can't, let's say, work or take a day off or have to retire early with disability, it adds up to almost $140 billion a year in the US. Not just US-wide, we think of ourselves as being a pretty healthy state. 6% of Californians also report significant visual impairment. So we have a number of really big unmet needs in ophthalmology. We need better measures of disease. These are called biomarkers. These are the ways that we tell whether you have a disease and whether it's getting worse or staying stable. We need treatments to prevent disease, and those could be drugs, those could be surgeries. We also need treatments not to just prevent disease, but to restore vision. So many of these eye diseases lead to irreversible blindness. And so we want to figure out not just neuroprotection, but how do we promote regeneration or even enhancement of vision? And how do we bring biomedical and bioengineering innovations to that task? So we're quite, as I said, I'm quite excited about how these are going to come from Stanford. And in fact, I can announce to you today, and we'll have a press release broadly soon, but you guys get to be the first to hear that we're initiating the new Mary and Sash Spencer Center for Vision Research here at Center, where we're going to bring together and consolidate vision-related research engineering, neuroscience, gene and cell therapies to hasten this translation from basic discovery into clinical care. So let me give you a little bit of a flavor for some of the advances that we're making. Let me start with the biology, some of the advances that we're making in the discovery. Now, everyone always is asking about stem cells, and they come into the clinic, well, what about stem cells? Should I get stem cells? So what about stem cells? So if here's a diagram of our retina, and here might be the normal complement, in this case of retinal ganglion cells that die off in glaucoma, stem cells can do two things. One is early in the disease, it turns out that they're little factories, and they can make all sorts of good survival and growth factors that can tell your remaining retinal cells to not die off in disease. And so they might be quite good at preventing vision loss in these diseases, and that in fact is moving into clinical trials right now. Even more exciting is the idea that the stem cells could turn into the cells that you've lost and actually replace or repair the damage and restore vision in these diseases. Now, this is moving forward, and I'll tell you it's moving forward quite quickly for the rods and cones in diseases like macular degeneration. We'll come back to that at the end. It's a tougher deal on the retinal ganglion cell side because not only do they have to integrate into the retina, they have to grow all the way back to the brain. Otherwise, they're not carrying your vision back to the brain, and they're not helping you see better. But we've had some very exciting advances this past year. These are actually transplanted retinal ganglion cells in a rat model of the eye. And these green cells are all of the donor cells that we've injected into the eye. And what we've found is that they integrate into the retina and they respond to light. And not only do they do that, but they grow all the way back to the brain. These are cross sections. Oops, these are cross sections of the brain. So they grow all the way back to the brain, and they go to the right targets in the brain. So we're now growing donor cells to grow all the way back to the brain and to replace the retinal ganglion cells that we know degenerate in glaucoma. So that's very exciting. How about gene therapy? One of our new faculty members we've just recruited, Vineet Mahajan, he's a retina clinician scientist and specializes in retinal degenerative diseases. And he's the first person to use a new kind of gene editing. This is actually like kind of a scissors that goes into your genome and can cut out the bad genes and put in the replacement good genes. And that's a new technology that's very exciting right now called CRISPR gene editing. And this CRISPR gene editing, we're very excited about repairing the gene deficits that sometimes lead to these degenerative diseases. We've done a similar kind of gene therapy to actually improve optic nerve regeneration. So here the eye would be on the right side, and normally if the optic nerve is injured, there's no growth down the nerve. Well, we figured out a gene therapy called KLF9 knockdown that allows these fibers to grow all the way down the optic nerve and in fact cross over into the brain. So very exciting advances on promoting regeneration. This optic nerve regeneration, Andy Huberman, one of our faculty here at Stanford, showed this year that if you combine that kind of gene therapy that promotes survival and growth with special kinds of visual stimulation, where you actually send specific kinds of visual stimuli into the eye, it can greatly promote that kind of optic nerve regeneration even further. And I'll mention again at the end, I hope, but we're actually moving this with these virtual reality goggles into human testing now as well. We can label up and mark all of the different cell types in the retina with fluorescent proteins that let us see inside. And Sui Wang is actually figuring out how all of those cells respond to diabetic retinopathy so that she can discover the next generation of therapies. So lots of exciting advances on the biology side. How about the engineering? Stanford is known, of course, for its biology, but also for its engineering, where we have one of the best engineering and bioengineering departments in the country. And innovation in this space has been really exciting in ophthalmology because we're a field of early adopters. Ophthalmology is actually the first area in medicine that lasers were used. The first tissue transplants were done in ophthalmology. And so it's a very innovative field in that way. Daniel Palanker is figuring out the laser retinal tissue interactions. and optimizing those for new types of surgeries and new types of therapies. He's also figured out how to make special silicon chips. These are little computer chips that can be implanted inside the retina and basically give you a bionic retina. So for patients with macular degeneration or other related diseases, these are entering human trials this year. Here's a picture of the inside of a rabbit eye where they've put about, 7 of these inside right in the center of the rabbit's vision. And we can show that they're getting right under the retina and keeping things really healthy under there. And so very exciting that these will be able to absorb light and signal back to the rest of your brain the vision that you've been missing in the advanced stages of these diseases. Now, another really important recruit that we've just made in these last couple of years has to bring Dubra to the department. Dr. Dubra is the absolute world leader in optical engineering for how to image the structure and function inside the eye with unprecedented resolution. Alf. Yeah.
Thank you. Well, thank you as well, all of you, for coming. And so let me briefly describe. So if you were to go to the clinic today, the way eye disease is traditionally studied and followed up over time is by taking a picture like the one that you see here. And this picture is a little bit as if you were looking at the eye, at the retina, that is maybe like 1 inch by 1 inch through a magnifying glass. And that is good, but we want to do better. So we want to look at the retina as if you were looking through a microscope. And in order to do that, we recruited technology from the field of astronomy. So it turns out that the same technology that you could use to look at the stars when people talk about 30-meter telescope, so we're going to use exactly the same technology and the same ideas to look inside your eye to see the cells instead of the stars. So if you were to zoom in a camera that doctors have in a clinic today anywhere around the country, you would see a blurry image. But if you apply adaptive optics, you can actually see really sharply to the point that you can actually resolve individual cells. And each of these bright dots here are not stars. They're actually your pixels. Those are your cones and rods that Dr. Goldberg was referring to earlier today. One of the things that we discovered in our group recently was that in addition to looking at this kind of stars-like view on the left side of the screen, where you see some dark areas, those are faulty cells. And for the longest time, we couldn't tell whether the cells were there or not until we invented the technology to look and create images like the ones on the right side of the screen. And that thing that looks a little bit more like bubble wrap, each of those bubbles is a cell that is there, but it's not working. So this is really important because now we can tell the people that are developing the gene therapies in our department and other places in the world, say, hey, these people these particular subjects that have these cells there can actually enroll in the trials and benefit from the therapy. Because if you don't have the cells to begin with, then the therapy is not going to do anything for you. The gene therapies assume that you have cells that are broken so that you can go and fix them. So now we can inform doctors as who is a good subject for these trials. In fact, we're hoping to do more than that, and we're doing this with collaborators here and worldwide. That the idea is that we're not going to only help recruit the patients, but also follow them over time. Because one of the problems that people face all the time when developing new drugs or new treatments, whether it's for vision or for other problems, you have to wait years and to monitor the changes, the improvements that the drug might be doing over time to see if the drug is working or not. So by looking at this tiny, tiny scale, we're hoping to learn a lot faster if the therapy works or if it doesn't work, so that we don't continue spending effort and asking more people to enroll in the trials if the therapies are not working. And if they are working, we can get them onto the market faster so that a lot of people can benefit from the therapies earlier. In fact, going back to the analogies of the stars, it turns out that some of the cells in the retina twinkle just like the stars. And one of the things that we're trying to explore is in the same way as you have an EKG of your heart and see how your heart is doing just based on how the electrical signals change over time, we're trying to develop, let me hopefully play this again. Anyway, so if we can monitor this over time, we're hoping that we're going to be able to tell you how every cell works, and if the therapies are working or not before you even lose them, because that's the important thing. That's the important thing we want to avoid, that you lose a cell. So if we can tell that a cell is sick, we're really moving far ahead. And then one other front that there's a lot of excitement in the field is because over the last few years, there's been a drug that has been transformative for age-related macular degeneration and other conditions that make blood vessels leak into the retina, is that we have now the methods to see not only the smallest vessels, but even the finest capillaries. In fact, they're so small that for those of you who might have seen a picture of a red blood cell or a diagram of a red blood cell that looks like a red donut, the cells actually have to fold onto themselves to squeeze by. So they're really, really small. So now by using this adaptive optics technology, we can now see the smallest defects. And that is important because now, for example, you can see in as little as a couple of weeks whether a treatment is working or not. And let me show you here how there is this little gap in the blood vessel that 2 weeks later is actually letting blood flow through it again. And in fact, here, the arrow seems to point to nothing, and they actually do point to nothing, so don't look too hard. And there is a blood vessel here that is not that it grew in 2 weeks, but it was there to begin with, but it wasn't working. So now the doctor can tell the patient, hey, this treatment is working, or your restriction of sugar is working. So rather than having to tell the patient, well, but the patient might sometimes say, oh, I don't see any changes, so why should I continue treatment? But now we can help both the doctor and the patient to keep motivated and continue treatment. In fact, we invented a technology that allows us to see these things that look really grainy, but they look like really fast moving sausage-like things. Those are the red blood cells. So one of the things that we're going to do is to develop a machine to measure the velocity of each of them, so we don't have to wait until your blood vessels are blocked, but rather tell you, hey, the blood is slowing down over here. So this might be an even early damage. So again, the ultimate goal is to treat so early that you never experience vision loss. And earlier today, Dr. Goldberg mentioned that one of the most prevalent conditions in the developed world is glaucoma. And we are developing methods to monitor how the cells actually transport nutrients and mitochondria along the axons. And we're going to try to monitor the twinkle, because it turns out that there's more than just the photoreceptors that twinkle, to see if we can detect early signs of glaucoma by monitoring how each of these little bright dots actually twinkles over time. And in fact, if you were to look at the center of each of these kind of movies here, you might see that there's something that looks like a worm that's kind of moving along. And these, we're hoping to confirm this, but we think are mitochondria. And these are really essential components of the cells that provide a lot of the energy. And if these mitochondria are not moving along the axons, then the cells are not very healthy. And one of the things that we also do, and we always argue that this is the right thing to do, both for the NIH dollars that are invested in research and also foundation and donors' money, is that we share a lot of the technology that we develop so that we can maximize the impact. We think is the right thing to do. And we've shared, we've actually built machines in other places, so we spend a lot of time in airports and in other places just commuting. So we're obviously here, but we've gone as far as Norway. And we also have shared software, and we train people from other institutions so that they don't have to really reinvent this technology. And we can all together as a community move faster. So with that, we continue with the biology.
Wonderful.
Thank you.
I can't overemphasize what an enormous leap forward Dubra and his colleagues are bringing to ophthalmology and to the opportunity for precision health and for therapeutic development, because our ability to measure a disease, it's one thing to have the biology, but you put the biology together with this kind of advanced engineering, and it allows us to do what we call translational research at an unprecedented rate and level. And there what we're doing is we're designing the trials that will allow us to move the therapeutic candidates out of the laboratory and into the clinic. So how will these better measures, how will these better biomarkers that Dr. Dubra and others are creating lead us to cures for diseases? Well, you know, a lot of these diseases that we're talking about, patients kind of decline fairly slowly. And if we're not that good at measuring the diseases, it can take us a long time, sometimes years, to really know if you're kind of staying about the same or getting worse. You know, we're often looking back over the last couple of years to be able to tell our patients if they're getting better or staying about the same, getting worse. But with a better and so that takes a long time to study. And so if you want to do a new therapeutic trial, that means it's a long trial, and that's hard, it's laborious, it's expensive. But if we have a better biomarker, a better way to measure the disease and say, hey, if we give you the therapy and we can tell the next week that the cells inside your eye are feeling healthier and more robust, that's a huge opportunity to say, well, the drug must be working. And so that allows us then to measure in a short period of time and test new therapeutic candidates more quickly. So what are some of these new therapeutic candidates? Well, we're fortunate at Stanford to be really at the vanguard, at the leading edge of a lot of new therapeutic trials. We have new trials in glaucoma right now. For example, we're inserting a little implant, a little 1x5 millimeter tube inside the eye that actually, it's a little factory. It makes a drug called CNTF, ciliary neurotrophic factor, and it makes it inside the eye for years. And this drug has been shown to get the axons in the animals to regenerate all the way back to the brain. And so now for the first time, using advanced measurement techniques, we're seeing whether we can restore vision in glaucoma. I mentioned those little microchips that can go underneath the eye that Daniel Palanker and his colleagues have invented over the years. And this is also entering human trials this year. And what will happen is the patients will wear these special goggles that will have video cameras that absorb the light of what you're seeing around you and convert them into special light signals that go right into those little bionic chips in the back of the eye. And we're going to see how much vision we can give back to patients now for the first time. Let's see. Here we are. Daniel Palanker has also figured out a way to use lasers in a way that aren't damaging. So we heard earlier about a new treatment, very important for wet macular degeneration. Right now, there's almost nothing really for dry macular degeneration. That's the more common form. And what happens in dry macular degeneration, in part, is that these little accumulations or concretions, they're called drusen. They get underneath the retina and they kind of damage those rods and cones and keep them from functioning properly and lead to more trouble in the retina. And so by using actually a special kind of laser therapy that's non-damaging, it actually just tickles the biology of the cells in the back of the eye and convinces them to kind of clean up the extra fluid that's accumulating under the retina in some diseases, or clean up those drusen perhaps. So this again is in clinical trials at the Byers Eye Institute right now. Here's an example of a patient who actually had a fluid accumulating underneath the retina and was seeing 2100 on the eye chart. And 1 month later, 2 months later, that fluid has completely gone away from this non-damaging retinal laser therapy, and the patient is now seeing 20/20 in that eye. Very exciting steps forward. How about for the patients who have lost a lot of those cells? You know, they've had the damage for a long time. Those are the patients, remember, we talked about who are probably going to need stem cell therapy. And Ted Lang, one of the faculty in the department, was actually the first person in California to implant candidate therapeutic stem cells in a stem cell trial underneath the eye. And the idea here again is that in macular degeneration, you're losing all these rods and cones, but by putting the stem cells in, it can protect them and make them all healthy again. And right now, the kind of next generation of stem cells for these diseases like macular degeneration is being refined and gotten ready to move into this next sort of version 2 of therapeutic testing. So with that, I know we wanted to save time for questions, and I just wanted to point out that, again, we're making huge steps forward as a field, and particularly, of course, we're proud of the steps forward that we're contributing here at Stanford on the discovery side, on the engineering side, and of course, on the translational side, bringing new therapeutics into clinical trials. And so, of course, people always want to know, how can we engage? Well, obviously, we want you individually to make sure your eyes are healthy. So please feel free to come out to the pavilion if you haven't already. We have an eye screening booth among the Health Matters booths out there. And of course, you can always come and see us at the Byers Eye Institute. You can sign up for appointment if you want to take a picture. There's the phone number there for the appointments, or you can visit us online at our website. It's easy to remember because it's just ophthalmology.stanford.edu. And if you want more information about learning about our research, Melanie Erasmus, who's right here in the front row with one of these red shirts, or any of our red-shirted volunteers here today can help take your name and we can follow up with you. So with that, let me stop there and open it up to your questions.
Thank you.
1 second.
A couple of questions. I had an iridotomy. Is that what it's called? Is that for back of the eye glaucoma?
That's for back of the eye glaucoma. The iridotomy is the glaucoma. One of the big risk factors for glaucoma is when the eye pressure goes up. And one of the ways the eye pressure can go up acutely or all of a sudden is if your iris plugs the drain of the eye. If it drifts forward inside the eye a little too much, it can plug the drain. And to prevent that from happening, we put a tiny little laser hole in the very edge of the iris so it won't sort of balloon forward. And so that's called an iridotomy, yes. And it's very effective in preventing acute angle glaucoma.
Second question. My husband recently had a detached retina.
Yeah, yes.
And he had that gas
bubble, yeah.
bubble.
Yeah.
How long until he knows how his vision will end up?
Well, oh yes, thanks. I was reminded to repeat the question. So if you have a retinal detachment and you get treated either with surgery or surgery with a gas bubble, or occasionally you could have a tear with just the gas and treat with just the gas bubble. When the retina reattaches, and your retinal surgeon can tell you how that's coming along, because we can use some of this advanced imaging to show that. When the retina reattaches, sometimes the vision comes back right away, but sometimes it actually takes a while, and it can take even up to a year to see whatever the full benefit of that retinal reattachment might be. Interestingly, the same non-damaging laser that can tickle the cells in diseases like macular degeneration, we actually think that it could also tickle the cells in situations like repairing a retinal detachment and get them to fill in the gaps, fill in the holes of the vision that might be left over after that disease. So, that would definitely be a great question to bring to one of our retinal specialists over at Byers who are participating in that trial. And that could be, for example, Ted Lang or Steve Sanislow. Yeah, he had his hand.
Thank you for a very fascinating presentation. My question is about the occipital lobe. Visual impairment due to blood clot to the occipital lobe. Can you
talk about that? Sure. He asked about the occipital lobe, and what about the vision and if you get a blood clot or a stroke of the part of your brain, the back part of your brain is the first cortical way station of all your vision, and a lot of processing happens back there. So if you get a stroke that hits the back part of the brain, it's going to affect your vision. And actually, there's a fair amount of research using stem cells to fix those areas, but also using drugs that increase the plasticity. It turns out, when we're children, we're very plastic, and our brains can adapt to both our normal development, of course, as we learn, but also to our also to injury. And we lose some of that plasticity as we age, as unfortunately we're all too aware of. But it turns out that we're discovering the molecules, the genes that are important for that plasticity. And actually, Carla Schatz, who's a PhD vision scientist and the director of the BioX here at Stanford University, has discovered some of the molecules that are really important for the plasticity. And the idea that we could reopen plasticity in adults with stroke or adults who had amblyopia growing up, this is a very exciting area. It turns out that another exciting area might be that if we sort of exercise those parts of the brain, of the vision, for example, through specific types of visual stimulation, maybe we could also exercise the plasticity and improve the vision that way. So these are areas of very active investigation, both preclinically and also moving into human trials.
They used to have all these, there used to be these, which I thought were hoaxes, books and tapes on exercising your eyes to improve your vision. And I tried them once or twice and they didn't work, so I gave up on them. But are you saying that in fact there might be exercises? Do you think there was any validity in these old ones?
Yeah, so the question's about eye exercises. And you know, I tried running once or twice, and I didn't get in great shape either. So there's some good data about different types of eye exercises. There are certain eye exercises that help with your eye muscles and making sure your eyes stay straight so you don't get double vision in certain situations. There are other eye exercises that are being tested for the performance of your vision. For example, using glasses that cut back on your visual input and still make you practice doing certain visual behaviors. And so the idea is if you can do it with less visual input coming in, then when you take off those special glasses, maybe you'll perform even better with the full amount of light coming into your eye. So there's actually a fair amount of research going on in that area right now, and some of it is a bit questionable, as you rightly point out. But I think there is hope and plausibility and good biological evidence that kind of exercising certain aspects of our vision may help with plasticity or improving vision over time.
Then the next question, of course, is where do we get those exercises?
Yeah.
Are there any studies that are looking at it?
There are some studies, and I mentioned one of them that we're starting with the virtual reality goggles that are going to feed very specific visual stimuli and exercise your vision in a particular way. So that might be one approach. Some of the data, it's very hard to pick out, like if you just go on the internet, what's good and what's, you know, kind of a bit hoaxy. And so, yeah, so I appreciate that that's a challenge still today.
Anything in the future for epithelial membrane?
Yes. The question was about what about for epithelial? Do you mean epiretinal membranes? Yes. So epiretinal membranes are a scar that grow across the inner surface of the retina.
I thought it was a bump.
On the inside of the retina. It can make an actual bump, yes, and it can tug on the retina, make a bump or a pit. And those can be peeled surgically, although we usually wait for patients to get a little worse before we do it, because it's not like a simple surgery to get through. It's gotten a lot better now, so we actually will do it much earlier in the disease process. But that's actually one disease where we can fix that surgically. Now, there's almost no one anywhere in the world working on what's the biology, why does that happen, what cells are making that scar, and maybe we could develop a drug that could just prevent or even erase that scar. And I think that's a very active area. We have a couple of faculty who are quite interested in that area, but we really need to, that's an area of research that needs to be pushed harder, this epiretinal membrane formation.
Can central serous retinopathy turn into AMD later?
Can central serous retinopathy turn into macular degeneration? They're two different diseases, and they both can lead to fluid accumulations in the retina, but there's no evidence that the one causes the other. Central serous retinopathy is often caused by stress hormones, or if you're getting a hormone therapy for something else, like a steroid, let's say. So if you got really stressed about your macular degeneration, then that might cause central serous retinopathy. But other than that one exception, they're really two independent issues, and you shouldn't worry about the one causing the other.
The CSR doesn't cause AMD?
Right, it does not, it's not thought to cause AMD. Someone in the middle here. Oh, here you are, yeah. Oh.
Go ahead.
A question about the glaucoma and the cataract. How do you determine it's a time for laser surgery? And also, is it okay to have both surgery on the same eyes?
Yeah. The question is about glaucoma and cataract. Age is a major risk, increasing age is a major risk factor for both glaucoma and cataract, and it's quite common for people who have glaucoma and might be getting to a stage where they need surgery to also have gotten to the age where they might need cataract surgery too. And each patient, it's a little bit different, so that's really a discussion for with your surgeon. But I can tell you that it's quite common that if the patient has both problems in one eye, we can take care of both on the same day of surgery. And that's a very successful way to approach that. With the microphone, yes.
Yeah, thank you for your talk. My daughter has strabismus, and you know, when she initially, it was talked about as an eye muscle thing, and as time has gone on, it's been talked more about a visual development issue and a brain-based issue, but the treatment still seems to be surgery. And you were talking earlier about vision therapy. Can you just give me your perspective on it and where treatment is going at Stanford for strabismus and vision therapy?
Absolutely. So the question was about a strabismus, which happens in children, although also in adults. And that means when the eyes don't point straight together, if one eye drifts in or one eye drifts out, for example. And it's a particular problem in children because if the brain is not receiving visual information during development equally from both eyes, it'll start to ignore the eye that's not sending consistent information. And by a certain age, our plasticity that we talked about before, the brain kind of shuts off plasticity and stops caring about the weaker eye. And then that can leave children, or let's say by teenage years and into adulthood, people with permanent loss of vision in that eye. So it's very important that we sort of think about both problems. And what we do to treat strabismus and what's called amblyopia, which is the problem where the brain is starting to ignore one eye. So we want to line up the eyes again. So that's muscle surgery too. Even though it's not really the muscle's fault, it's actually the part of the brain that is controlling where the muscles point is probably just not doing that well enough. It's not getting the right kind of feedback loop going. But we can't go in and fix that little nucleus in the brain yet. So we fix it by fixing the muscles and giving them an advantage so that they yoke them straight. So we want to get the eyes straight again so the brain starts receiving equal information from both eyes. And also we need to treat the plasticity. And when children are younger, or even now it's appreciated into the teens, if you patch the good eye and let them see out of the weaker eye, their weaker eye will catch up to the good eye. And actually there's some really neat new versions of that where, for example, now listen, I have children and I'm always telling them to not play video games. But in this case, if you actually get both eyes to work together, not just looking at something passively, but having a motor output, for example, responding to that visual information, as one does when one plays a video game, it turns out that that actually reinforces the brain's reception of those two eyes' input coming in. And so there is some advanced thinking about that. Scott Lambert, one of our new faculty members in pediatric ophthalmology and adult strabismus, is really one of the national leaders in these kinds of questions and on the sort of vanguard edge of how to treat these patients and how to think about the visual development and the cutting edge of therapy. And so if you haven't met him yet, I would encourage you to meet him. Yeah.
research on nutrition as far as macular degeneration? I know my mother was diagnosed with it, and my optometrist said, oh, well, you should take these vitamins because this will help prevent it in the future because it's passed genetically.
Yeah, so macular degeneration, the question was about vitamins and nutrition for macular degeneration, and of course for other diseases. For macular degeneration, it has been shown that those nutritional supplement pills that you can buy over the counter in the pharmacy have been shown to prevent macular degeneration from going from the moderate middle stage of the disease into the severe stage of the disease. Because the vitamins are thought to be healthy in general, we actually usually recommend that even if you have the mild form, go ahead if you want, take the vitamins. This comes up a lot with other eye diseases like glaucoma, and for a lot of eye diseases, we haven't really been able to prove that one nutrition versus another, one diet versus another, one supplement versus another is really critical to preventing or treating the disease, say in glaucoma or some of the others. But we still, as doctors, always recommend, eat healthy and exercise, even if it's a brisk walk around the block for 20 minutes each day, because the things that are good for your blood flow and your heart conditions and keeping your cardiovascular system healthy, the retina and the optic nerve are very sensitive areas. And so anything you can do to maximize their oxygen consumption and oxygen flow and blood flow, we think that's going to be healthy. So we back up the generic advice of eat healthy and exercise. One over here?
Well, I really had a similar question to the previous regarding dietary supplements and prevention of eye diseases. So you're saying that with respect to macular degeneration, the supplements are useful. These are specifically, what are they? Lutein? And
yeah, lutein, zeaxanthin. They're called Ocuvite. There might be a couple different brands. They're over the counter, but you can ask the pharmacist to direct you. There will be 2 or 3 brands in one section of the that have the exact right. Let's say that have the exact combination that was studied and shown to be positive.
Great. And any research taking place at Stanford along these lines of dietary supplements and prevention?
Dietary supplements in all areas of research require clinical trials with often hundreds. In the case of the macular degeneration nutrition trials, thousands of patients, 4,000 patients across the world participated in order to be able to prove the effect of the nutrition. So it takes a lot to get that kind of trial off the ground. We're hopeful that with the development of new ways of imaging these diseases and imaging the retina and being able to look not just at the structure but also the functioning of your retina, that we could actually start answering questions like that very quickly. We could say, here, take this vitamin for a month, and we'll take special pictures of your eye again, and we'll tell you if your retina looks healthier. And so we'll know for you, for the individual, kind of a precision health approach, how you're doing.
Are there any preventative measures for preventing retinal tears?
Retinal tears, can you prevent retinal tears? Retinal tears are actually fairly common. Sometimes we discover them when the patient's not even symptomatic, just sort of, turns out you've had one and didn't even know it. They're more common as we age. They're definitely more common in people who are nearsighted, especially if you're fairly severely or strongly nearsighted. That's you? Okay, yeah, it's very common in our community. The truth is probably not. You could hypothesize that if you jiggled your eye around less, for example, you were not exercising and just sat very carefully on the couch, you might decrease your risk. But we've, of course, never studied that question. And we would still prefer that everybody exercise. So the answer is, maybe stay off the football team. But really, really there's not a great way to think about preventing those reasonably. Yeah, we would think that jogging would be safe. It's not like high impact, like being a football linebacker might be. Yeah, so maybe don't be the linebacker on the Niners. I know you were thinking about it, but.
was wondering if stents are still on the table for lowering pressure of glaucoma. Are they still being studied?
Yes, the question was about stents for lowering pressure in glaucoma. Remember, one of the big risk factors for glaucoma is increasing age, but the other is increasing eye pressure. And once we know you have glaucoma, we want to lower the eye pressure. And we have eye drops that can do it, we have lasers that can do it, and we have surgeries that can do it. Obviously, we usually like to do the surgeries later on if the others aren't working. And one of the ways these surgeries work is we can put in these tiny little stents that can actually open up a little kind of trap door that releases the fluid out of the eye. And obviously, if you let the fluid drain out of the eye, it doesn't go down your cheek, it goes back into your body. But if you drain the fluid out of the eye, then the pressure will be lowered. And some of the stents work really well if you have high pressure and we're trying to get you to modest levels, and others may work better if you have modest and we're trying to get you to really low, like if you have advanced disease, for example. So still very much an active area of investigation. Actually, Kuldev Singh, who's one of our glaucoma faculty here at Stanford, and is really one of the internationally prominent glaucoma faculty in the world, he's actually has a collaborative grant from the CDC to actually determine what are the outcomes of these different kinds of surgical procedures, and also what's the patient satisfaction with these different procedures. So looking at comparative effectiveness and patient-reported outcomes is a very important area in medicine in this next generation of medical studies, and he's really leading that for glaucoma.
Hi, I have a question regarding contact lenses use. I work in front of a computer 8 hours doing data analysis, and I still wear a contact lens. Should I be worried about the oxygen? And in the long run, is there any data suggest?
Sure. So the question was about wearing contact lenses, and especially if you're in front of the computer all day, which is also very common in our community, right? So contact lenses are generally safe. The most important thing about contact lenses is actually being really good about cleaning them, or elect to use the dailies version, which are a little more expensive, obviously, but then they're really safe because you're not worried about getting an infection from inadequate cleaning. The oxygen actually flows through modern-day contact lenses quite well. You'll know if you're having a problem. You won't tolerate the contact lenses if you're having an oxygen problem. The other problem, more commonly, that people have with contact lenses, especially with people who are in front of the computer a lot, or maybe watch a fair amount of TV or love to read, is that when we do those three activities, we blink less. And when you blink less, your eyes dry out. And when your eyes dry out with contact lenses, it's particularly uncomfortable. So whether or not you wear contact lenses, if you're in front of the computer a lot, help yourself to an over-the-counter bottle of artificial teardrops and use them throughout the day and keep your eyes moist, and take little breaks, even if they're not long breaks. Take little breaks from the computer where you look around the room or look out the window or something like that. Yeah.
Yes, is the eye pressure the only cause of glaucoma, or there are some more other factors involved? And even though you do the eye drop, you lower the eye pressure to very, very low, but this thing can continue to degrade. And is there any breakthrough and study in those areas?
That's a very important question. The question was, we know about the eye pressure being a cause of glaucoma, a risk factor for glaucoma. Well, what else do we know about that, or is there anything else? Because sometimes we lower the eye pressure and the patients still get worse. We know, because that happens, that there's a significant part of this disease that is not just due to the eye pressure. Something else. There's some sensitivity or susceptibility in certain people that their optic nerves degenerate in this glaucomatous fashion. And as a field, we've really tried to dig into that question. And we know there are some what we call minor risk factors, like sleep apnea, and so you can get yourself checked for sleep apnea. There are other examples of kind of minor risk factors. So we've tried to dig into that question on the research side, particularly over the last few years. The field has gotten together to try to figure out, is there a gene or is there some genetics that we could figure out for glaucoma that would lead us down a more fruitful path? And with, in different studies, 6, now 12,000 patients participating in genetic studies, we still haven't found a really strong gene like we have for macular degeneration or some of the other diseases. There are a couple of genes that are probably significant players in a small number of glaucoma patients, or maybe have a very small effect in a broader number, but there's no slam dunk on the genetics of glaucoma. We're hoping that this advanced imaging kind of approach is going to lead us into a better understanding of what's really happening in the disease. And the thinking is that if we can discover what's happening in the early stages or the progressive part of the disease, that then we can design therapies that are maybe not about the eye pressure. Part of that has to do with the neuroprotection and neuroenhancement trials. So for example, that little implant that I mentioned earlier, a little 1 by 5 millimeter tube that makes a drug inside the eye, we put it inside the eye and it makes it for years. That actually has nothing to do with treating the eye pressure. It is purely a drug targeted at keeping the retinal ganglion cells alive and getting them to grow back down the optic nerve and function at a higher, more robust level, kind of enhance their function. So, so we're testing actually for the first time really recently new therapeutics that are not about the eye pressure for glaucoma, but are about the degeneration, to ask if we can prevent the degeneration or even restore vision. There's another one we're doing right now. It's actually delivered as an eye drop, and it's a related factor called nerve growth factor. And certainly, if you're going to get treated with something, nerve growth factor sounds like a good name for it. So anyway, this nerve growth factor, we're testing with a topical therapy, but it penetrates into the back of the eye, and we're seeing whether it can enhance the function, enhance the vision of glaucoma patients. So that trial is just opened up right now. We'll take maybe 1 or 2 more, and I'll mention that we'll be, I'm happy to stay afterwards, but.
Can you tell me about floaters? And is it caused by cataract surgery?
Yeah. Floaters, the question was, what's with these floaters, right? We often see them, right, floating around, and they'll come and they'll go, or maybe you'll have a bigger one that really gets in the middle of your vision and annoys you for some extended period of time. The middle of the eye is filled with jelly, and as we age, the jelly kind of shrinks, and it's actually that shrinking that pulls on the retina and creates a retinal tear that came up earlier. You know, not in everyone, of course, but in a small number of patients. As that jelly shrinks, it kind of gets little concretions or little pieces that are floating around in there, and that's what you see as floaters. And some will come and some will go, and we don't really have a really great treatment for them. Now you asked about, so what we really actually advise is just ignore them, and your brain will start to ignore them. Not that satisfying of an answer, right? Yeah. Yeah. Ignore them and they'll go away. You asked about cataract surgery. Cataract surgery doesn't cause floaters, but when your vision improves after cataract surgery, you can see them better.
My ophthalmologist wanted to lower my eye pressure, IOP, and so he drilled some holes with laser.
Yeah.
And I'm wondering, can it get too low, and what would be considered too low?
Yeah, the eye pressure can, in theory, get too low, and in theory, just like a balloon, it could go to 0. Sometimes, very rarely after glaucoma surgery, one of the risks of glaucoma surgery is, well, what if it goes too low? Usually the lower the better, so frankly, if it's even a little too low, we're all pretty happy about it. It really probably with the laser can't get too low. With most of the kinds of glaucoma lasers that we do, there's probably not a significant risk there of going too low, but some of the surgeries it can. If the retina gets too low, you know, if a balloon gets really low pressure or out of air, it wrinkles, right? It's not puffed out, it's kind of wrinkled. If the retina in the back of the eye or the cornea in the front of the eye wrinkle a little bit from the pressure being too low, you know, that can degrade the quality of your vision, because instead of looking through a clear pane of glass. Now you're looking through a wrinkled pane of glass. Things are going to look distorted. So too low pressure can interfere with the vision, and that's something that we keep an eye on. Luckily, after surgery, if you're one of those few people that happens to, we can go back and fix that. Yeah.
Would 12 be too low?
No, 12 would be wonderful. In general, we're so grateful if we can have a patient at 12. So we'll make an assumption that that's you and you're in good shape right now. So yeah. Well,
thank you. Please join me in thanking both Dr. Goldberg and Dr. Dubra.